Welcome to Produce Talks.
This whole business is based on relationship and friendships.
Food is darn simple.
What can we do? What are other countries doing?
How important it is for industry to be talking about this.
We're going back to the farm. We're going back to the whole food.
Hello and welcome to Produce Talks, the CPMA podcast. This episode, we feature a recording from this past spring during the CPMA Fresh Week, unpacking food safety and providing public health perspectives on emerging pathogens. Foodborne illnesses continue to be a source of concern within the fresh produce industry, and this panel featured two top food safety experts, Joyce Cheng, Manager Outbreak Management Division at the Public Health Agency of Canada, and Dr. Lawrence Goodridge, Director of the Canadian Research Institute for Food Safety at the University of Guelph. The panel was moderated by Lisa Sparrow-Moellenbeck, Food Safety Manager at Federated Co-operatives. We'll get you right to the panel after a quick word from our sponsor, Nature Fresh Farms.
Hey, it's Sarah from Nature Fresh Farms. Food safety is important to building trust with customers and ensuring consumer health and safety is preserved. At Nature Fresh Farms, we believe it's the responsibility of our entire team to support the development and implementation of food safety systems and standards. Explore our food safety practices blog post at www.naturefresh.ca/blog.
Hello, everyone, and welcome to Fresh Week Learning Lounge, Emerging Pathogens, a Public Health and Research Perspective. My name is Lisa Sparrow-Moellenbeck, and I'm the Food Safety Manager at Federated Co-operatives Limited, and I will be your moderator for today's session. Foodborne illness continues to be a source of concern within the fresh produce industry, and today we've brought together two of Canada's top food safety professionals to join us in discussing recent outbreaks, the challenges in identifying outbreaks, and what the future of outbreaks may look like. Our panelists today include Joyce Cheng, Manager of Outbreak Management Division at the Public Health Agency of Canada, and Dr. Lawrence Goodridge, Director of the Canadian Research Institute for Food Safety at the University of Guelph. Thank you both for joining us today for this important discussion. Before we begin, a quick note to the audience that this session is being pre-recorded, and we will be joining you live at the end of the recording for an audience question and answer time. Please feel free to submit your questions in the chat box throughout our discussion, and we'll aim to cover as many of the questions as we can following our panel. So to start us off today, Joyce, can you provide us with an overview of recent produce food safety outbreaks, and in particular those that were surprising to the agency?
Sure. So when I think about recent produce-associated outbreaks, there are three major themes that come to mind: E. coli in leafy greens, novel food sources, and cyclospora in imported fresh produce. So I'll start with E. coli in leafy greens. So E. coli 0157H7 outbreaks linked to leafy greens typically occur sometime between late summer until December in Canada. And we still see new cases with E. coli strains that have been linked to past leafy green outbreaks. So we're always monitoring the data closely to detect and respond quickly to the next leafy green outbreak. For 2020, we didn't experience any large E. coli outbreaks in Canada. And we also know that there were some import requirements in place for romaine. But we know the U.S. investigated a few E. coli outbreaks related to leafy greens. So the potential for outbreaks in Canada still exists, given that we import some of these leafy greens from the U.S. into Canada. The second major theme I wanted to talk about was that of novel or uncommon food sources. So last summer, we investigated two Salmonella outbreaks that involved a novel or uncommon food source. The first was a Salmonella outbreak that was linked to onions imported from the U.S., and that caused over 500 reported illnesses in 7 different provinces. And this was a novel food vehicle for us in Canada, and the high case counts actually made this the largest Salmonella outbreak in recent Canadian history. And I think that detail was sort of lost within the pandemic. Secondly, we investigated a Salmonella outbreak linked to peaches imported from the U.S. We knew from the 2014 Listeria outbreaks linked to stone fruits that bacterial contamination of peaches was possible. But last year's outbreak was the first time we actually saw an outbreak in Canada that was related to peaches. So that was new and interesting for us as well. The third theme that I wanted to talk about was Cyclospora and imported fresh produce. So Cyclospora infections in Canada are typically associated with either travel to countries in subtropical or tropical areas or consumption of fresh produce imported from these countries because the Cyclospora parasite is endemic in these countries. In Canada, we typically see locally acquired Cyclospora infections in the summer months. And at the Public Health Agency of Canada, we've been coordinating outbreak investigations of Cyclospora annually since 2013, and case numbers have been rising steadily every year. For 2020, what stood out was that we had almost 400 cases of Cyclospora, 399 to be exact, and that's by far the most we've had in any year that we've coordinated Cyclospora investigations. We were able to identify the likely source of some of the illnesses this year, and that included certain salad products, which were distributed in both Canada and the US. We also had some linked to imported fresh cilantro and fresh parsley. And we also saw some encouraging progress on laboratory sequencing methods for Cyclospora. And, I look forward to a time when we can make use of this laboratory tool to help with hypothesis generation in real time. So those would be the three major themes in recent produce-related outbreaks for me.
Thank you. And so, Lawrence, from a researcher's perspective, how do you determine which are the key pathogens to focus in on, and what tools have been developed recently to help with those investigations?
So Joyce has actually provided an excellent introduction to the answer to this question. Basically, the way that researchers decide which pathogens to focus on is really based on the ones that are causing the most outbreaks and potentially the most illnesses. And so Joyce has highlighted, for example, Escherichia coli and Salmonella, which in recent years have caused quite a number of outbreaks and illnesses linked to fresh produce. And I would also add to that Listeria-Moellenbeck. So those would be three bacterial pathogens that would be certainly of importance in terms of foodborne outbreaks linked to fresh produce. And then some emerging pathogens. So Cyclospora, again, has been mentioned. With respect to tools that are emerging, again, Joyce has introduced the concept of whole genome sequencing, which is certainly becoming very important with respect to foodborne outbreaks linked to fresh produce, both from a foodborne outbreak investigation point of view, where isolates that come from fresh produce can be now increasingly definitively linked to isolates that have been obtained from samples from sick individuals. That's important because then we know for sure that the source of the outbreak is a fresh produce vehicle, and that therefore then opens the door for us to do further investigations to figure out why that happened and hopefully stop such outbreaks from happening again. With respect to research on the pathogen side, genomic sequencing is also useful because by determining the sequence, the DNA sequences of these pathogens, we can then interrogate those sequences to ask questions regarding why these pathogens seem to consistently contaminate fresh produce and why certain pathogens seem to predominate. For example, E. coli in romaine lettuce. Why is it that E. coli is always seems to be the pathogen and not other pathogens like Salmonella, for example? And so by looking at the DNA sequences, we can gain clues into why some pathogens seem to persist in certain environments over others. We can also use that information to determine things like sanitizer resistance, which might explain why, for example, Listeria-Moellenbeck seems to be so good at persisting in food processing environments. And if we do see such sanitizer resistance, we can do things like change the types of sanitizers being used and so forth. So genetic sequencing, whole genome sequencing is really emerging as a key tool, both in terms of foodborne outbreak investigation and as well as on the research side, understanding why these pathogens persist in the environments that they do.
Great. Yeah, a lot of work being done. That's great. So maybe now from both of you, from the public health and the researcher perspective, what advice would you give producers and those along the supply chain to reduce the risk of an outbreak occurring or expanding? Joyce, maybe you can start.
Sure. So with respect to specific advice for producers and those along the supply chain, I would defer to the expertise of the Canadian Food Inspection Agency for that. But what I can say from the public health perspective is that produce-associated outbreaks can be very challenging to investigate because there is a reporting delay from the time when an individual becomes sick to when they're confirmed to be part of a national outbreak. And this time lag can take anywhere from 4 to 5 weeks on average. So as you can imagine, during this time, the food that is the source of the outbreak is either fully consumed or, it goes bad, leaving nothing available for testing, or the packaging, if there was any to begin with, is thrown away. So we lose that useful brand or lot code information that is typically helpful for doing traceback investigations. So because produce has this short shelf life, timely investigations are critical, and having that good traceability can reduce the time it takes to identify the specific food that's causing illness and have it removed from the market so that we can prevent the outbreak from expanding. Also, over the past year, we've seen a real expansion in the options that people have to purchase food beyond your brick and mortar grocery stores or restaurants. So during the pandemic, we heard a lot about, the food service sector offering groceries, also online food order or delivery apps, meal kit delivery services or subscription boxes. So as public health investigators, it's important for us to, learn from the industry what the latest trends are, because when we interview our cases for an outbreak, we want to make sure we ask about the full breadth of food supply possibilities to get a very comprehensive food history and make sure we don't miss anything.
For sure. Lawrence, any advice from you?
I think just trying to, first, understanding that foodborne outbreaks do occur and occur with fairly relative frequency in fresh produce. I think that has to be said. And I say that because while there certainly are a large number of producers who understand that, I think there are still some who would push back on that. So then with that information, the next step is to really make sure that each individual producer really conducts an audit of their production and their facilities. And so I say that because one of the challenges, I think, with fresh produce production is that unlike a ready-to-eat food that would be like a deli meat or something that would be produced in a food production plant, most of our fresh produce is grown outside. And due to geographical diversity, where some fresh produce is grown versus where other commodities are grown can be very different. So the point is that when we talk about food safety, we can't adopt a one setting fits all type of situation. We see this, for example, in the Western United States, where some fresh produce growing fields are right across the street from confined animal feeding operations, which have been linked to outbreaks, whereas other fields may be far away from that. So putting in food safety interventions that might work for one field might not be the same for the other. So conducting a self-audit of the production facility is and the growing environment is important. And just keeping abreast of new research with respect to how one can reduce pathogens. Certainly there's been a focus on irrigation water and how irrigation water, particularly surface water sites, can be treated to reduce the potential presence of pathogens. There's potentially some new diagnostic technologies that will allow one to specifically search for foodborne pathogens in the environment that is associated with the fresh produce growing environment, as opposed to looking for generic indicators like generic E. coli, which have not really been helpful with respect to determining the safety of an irrigation water source, for example. Finally, there's some emerging technologies that are being developed to actually process the fresh produce in such a way that it would reduce the presence of foodborne pathogens while still maintaining the fresh nature of the commodity. So, my advice would be for growers and processors to really keep abreast of these exciting discoveries and inventions that researchers are developing.
Great. That's some great advice. Thank you both. Okay, so here's the dreaded what keeps you up at night question. But what keeps you up at night when you're thinking about produce and the inherent risks associated with products that generally don't have that kill step in their process? Joyce, we'll start with you.
So for me, it's about finding the right balance between health protection and health promotion, because we have a mandate as an agency to prevent chronic disease and infectious diseases. So on the one hand, we have the risk associated with potentially contaminated produce that's consumed raw, which can lead to some, mild illness, hospitalization, and in some cases death. But we also recognize the benefits of eating fruits and vegetables to reduce risk for chronic disease. So when it comes to issuing public health advice for Canadians during an outbreak, I'm always thinking about how we can provide Canadians with the best advice to handle, prepare, and store fresh fruits and vegetables, while also enjoying the many health benefits that come with eating these foods.
For sure. And Lawrence, what keeps you up at night?
For me, what keeps me up at night is the fact that despite advancements in research tools like genomics, despite our increased understanding of how bacteria contaminate various ecological niches, we still do not have a very good understanding of why outbreaks are happening, certainly with some fresh produce commodities. So the example that I will use is E. coli and romaine lettuce. We know that potentially contaminated irrigation water could be a source. We know that cow manure is a source. We know that runoff during extreme rain events could wash contamination to a field. We know that wild animals could be a source. And yet, for most of the outbreaks that have occurred recently, we cannot definitively find a source. And that keeps me up at night, because if we can't definitively identify a source of the contamination and a cause of the outbreak, then we don't know where to focus our interventions. And so we as researchers, we need to continue to develop better methods that we can use for investigative purposes to try to definitively identify where the contamination is coming from, and why, for example, if confined animal feeding operations are a major potential source of contamination, then why do we see outbreaks with some growers that are in close proximity to those operations, but not with others who are in close proximity? So we really need to have a better understanding of that, and that certainly keeps me up.
Yes, for sure. So, and then finally, we're already seeing how climate change is having an effect on our primary production communities in areas such as maybe pest migration northward and unpredictable weather patterns. From a food safety perspective, are you seeing any evidence of changing or new pathogen trends in respect to fresh produce?
So perhaps I'll start. So my colleagues at the Public Health Agency of Canada have done some activities in this area through the FoodNet Canada program. And this is a program that does continuous and episodic surveillance activities at 4 sites, Sentinel sites across Canada. And they collect information across 4 components, human, retail, on-farm, and water. And this surveillance occurs throughout the year to identify trends in disease occurrence, different sources of exposure, and attributes illnesses to sources and settings for targeted enteric pathogens. So as part of the retail component, the program actually does some targeted surveillance activities that focus on select food items that are likely sources of human exposure. And the focus can differ from year to year, and fresh produce has been included as a focus priority as part of these surveillance activities. And over the years, we've done surveillance on things like fresh and frozen berries, fresh herbs, fresh cut fruit, leafy greens, ready-to-eat slaws. And this is also, responsive to what kind of trends we're seeing within the outbreak. So, for example, if we're seeing novel food vehicles like peaches and onions and stuff like that, those are things that can be targeted for surveillance in the future. And these trends are also monitored over time, so we can see how something like climate change could have an effect over time. So all produce that were tested for parasites, viruses, and bacteria, and generally the results yielded low recovery rates of pathogens through this program. From an outbreak perspective, we also have frequent communications with our partners in the U.S. to keep an eye on outbreaks that they're seeing and food recalls that are happening south of the border. And we know that there's a lot of import of produce into Canada from the U.S. So this gives us a sense of what's possible as far as novel food vehicle pathogen trends, and the possibility that we can have an outbreak here due to that source at some point. So we're always trying to keep an open mind about all the different possibilities when finding a source to an outbreak. And as we saw with peaches and onions, there's a first time for everything.
And Lawrence, anything to add?
Yeah, so I think we are starting to see some of the effects of climate change with respect to foodborne illness, and certainly with respect to some of the pathogens that have traditionally caused outbreaks linked to fresh produce. So there have been several studies in the U.S., for example, that have shown that there's a seasonal trend that seems to emerge with respect to the increase in infections, for example, caused by salmonella. And that seasonal trend seems to be linked to increases in temperature, in air temperature, where an increase of as little as 1 degree has led to increased cases of salmonella infection. So I think, as we look at climate change, I think we'll start to see, and as the planet warms, we could start to see increased infections linked to that. I note that with respect to outbreaks, we've also seen the potential evidence of climate change. There was an outbreak, I believe, in New Zealand a few years ago linked to melons, and it was thought that dust had spread the pathogen to the melons. And that dust, there were dust storms because it had been a dry year. There were drought conditions. So I think, again, drought being one of the things that we expect to see with climate change, that could increase how pathogens are spread. And certainly in some of the romaine outbreaks in the U.S., certainly in some of the outbreaks linked to romaine lettuce produced in the U.S., in the western U.S., dust and potential spread of E. coli, for example, from animal feces has been something that has been questioned and has been investigated. I also think that although there's no evidence to support this, we will start to see, novel food vehicles, novel fresh produce food vehicles be increasingly implicated in foodborne outbreaks because of the change in climate and because of what that will do to the pathogens and their ability to survive. So it's interesting to me that we saw the onion outbreak, we saw the peach outbreak, two foods not traditionally associated with foodborne outbreaks. And it'll be interesting to try to further study the parameters behind those outbreaks, and including the salmonella that was isolated to see if perhaps the bacteria have acquired an ability to survive in environments where they may not normally have survived, and also if those changes are due to climate change.
Yes, for sure. Lots of questions and lots to kind of watch for. Well, I'd like to say thank you to both of you for your insights on these topics and for such a really great discussion. Now we will join the audience for a live question and answer period. Good afternoon, everyone, and thanks for joining us. We are now joining live with our panelists for an audience Q&A. Listening to that discussion a second time, there was sure a lot of great information and a lot to unpack. So let's get to some audience questions. So our first question is, as a country that imports approximately 70% of our produce, what would the panelists suggest importers do to help mitigate some of the risks associated with imports? So I think probably that's for both of our panelists. So Joyce, if you want to start.
Sure. So I would suggest, or I would start by saying, know where your product comes from. That helps not only in an outbreak situation, when we do need to do traceback, having good records and traceability is certainly very helpful. But, also, do they have a good track record of putting out good product? Because we know that foodborne illness is not good for business. If they do have a track record of repeatedly being implicated in foodborne illnesses, there's a consideration there whether or not you want to continue a business relationship with them or take your business elsewhere with a company that has a better track record of producing clean, safe food that you will then eventually import and pass on to your customers.
Great, thanks. Lawrence, did you have anything you wanted to add to that?
Not really much to add to that. Just reinforcing, know who your growers and producers are. I note that the FDA recently, on April the 6th, released their final report with respect to an outbreak that occurred linked to romaine lettuce. And so they're now saying that E. coli 015787 should be a reasonably foreseeable hazard, and this idea that it comes from cattle. So I think one thing that importers can do in terms of when looking at producers and growers is to look for things that might increase the risk of a potential outbreak. So if for example the fields where the romaine is being grown or other leafy greens are being grown is immediately adjacent to a concentrated animal feeding operation, or there's one that's nearby, really making sure that everything possible is done to reduce the potential risk of contamination.
For sure. Some investigation needs to be done into the products you're purchasing. Another question that we got was, are there particular crops that are at a higher risk of novel issues? Not sure if you both want to take a turn at that. Lawrence, do you want to start?
Yeah, so that's a great question, and that's a question that I've been asked many times, and particularly recently, since 2017, I think there's been 9 outbreaks of E. coli linked to romaine lettuce. So the perception is that romaine is somehow, there's something, unique to romaine that's causing these outbreaks, and there could be, some certainly some of my colleagues have opinions about, the leaves of the romaine lettuce and how that might lead to more contamination. But overall, I think if we take a step back and look at the leafy green industry as a whole, we'll see that, since 2000 or so, there've been, outbreaks in many different leafy greens. So I like to say that it's likely not one specific type of leafy green or fresh produce commodity that's more susceptible. It really comes down to trying to control the sources of contamination that we know of.
Sure. Joyce, anything further?
From an outbreak investigation perspective, we're basically keeping an open mind. We know that novel issues can pop up at any time, and we want to be ready for it. So, there are your usual suspects, right? The leafy greens, we want to make sure we ask about that, but not to the exclusion of other possibilities as well.
For sure.
And I'll just jump in on that, and precisely because in the last year we've seen outbreaks linked to more novel fresh fruits and vegetables like red onions and peaches, which typically are not implicated in outbreaks. So I agree with Joyce. It's very important to keep an open mind when thinking about outbreaks and also contamination of different commodities.
Yeah, for sure. Lawrence, I'm going to ask you this next question. It says, because of the fact that there are so many unknowns overall relating to Cyclospora, what research or types of Cyclospora research do you think would be the best to fund initially?
Oh, that's a great question. I think we need to figure out where it's coming from. Because really, I think unlike Salmonella or E. coli, which, we have a pretty good idea in terms of animal sources, including wild animals, where it's coming from. I don't think we have a good handle on parasites like Cyclospora yet. So I would think that that would be a main cause of, or a main area that we should investigate, which is where is it coming from precisely? Because in theory, that should be easier to control than the bacterial pathogens like Salmonella and E. coli.
Great. Yeah. Joyce, you maybe had something to add into there?
I think one of the biggest stumbling blocks for us in the sort of the outbreak investigation standpoint for Cyclospora is unlike Salmonella and E. coli, which are bacteria, and we have well-established sort of sequencing methods to kind of delineate outbreaks from one another. With Cyclospora, we don't really have that. So we have a lot of individuals. We know that they all have Cyclospora, but are all Cyclosporas designed the same, and are they all necessarily part of the same outbreak? So there's definitely a lot of noise there, and without that additional laboratory ability to separate out unique groupings of Cyclospora infections, we have no choice but to investigate them all as a whole when we know that there's a possibility that they might not all be related to one another.
Great. This is another question that we got is fairly kind of relevant, and you both probably have some thoughts maybe. We've recently seen the rapid development of the COVID vaccine. So is there anything similar coming for pathogens?
That's a great question. Not that I know. So there are vaccines for some pathogens but those vaccines are to vaccinate like livestock, food animals, and so forth.
Yeah.
But not for humans, as far as I know.
It would be an interesting change in some of the with some of the outbreaks, wouldn't it, if we had some vaccinations? Another question that we received: are there any diagnostic or procedural changes on the horizon which might shorten the time between the onset of an outbreak and when it is identified in the public health agencies? So, Joyce, maybe you want to...
Sure. So there's things happening both on the laboratory side and on the epidemiology or the outbreak investigation side. So from the laboratory's perspective, so I should preface this by saying that the outbreak investigations and our ability to detect an outbreak is largely based on laboratory methods. So an individual will get sick, they provide a stool sample, the stool sample gets tested, and then that's where we find the bacteria within the stool, and then that gets sequenced, and then we identify that, they have salmonella with a particular genetic fingerprint. So all of that takes time. So from the time you show up to your doctor, it goes through multiple labs, it undergoes multiple tests, and it can take several weeks before we get to the point where at the national level, you're identified as being part of a particular outbreak. Some of the steps that have been taken from the laboratory perspective to shorten that time period involves decentralized testing. So when this whole genome sequencing method was first rolled out, it was led by the National Microbiology Lab, and they're based out in Winnipeg. So initially, a lot of these isolates were being sent there to be sequenced centrally. So all of that, shipping time to send these isolates to, Manitoba, it takes time, and it definitely added to the delay. What we've seen over the past few years is a decentralized model. So individual provinces and territories now have the capacity to do that sequencing themselves. So we definitely get those results faster. And that helps with the outbreak detection, right? Because then we have the results faster and we know when outbreaks are happening. The next one I wouldn't say is necessarily an issue right now, but it is definitely a consideration, is just around IT capacity. So whole genome sequencing is big data. We're talking about really large data files. So you need to have good, IT capability, strong servers, reliable IT systems to be able to move and process this volume of information. And then on the topic of a lot of the produce that we have in Canada being imported, and I alluded to it in my presentation as well, the ability to recognize that outbreaks are occurring in other countries is really important, especially when we think about produce. So, for example, an outbreak might be happening in the US, and we have systems that allow our laboratory databases to talk to the US databases and identify strains that might be showing up in both countries. Now, this might be an outbreak in the US, and in Canada, we might just have one or two matches that, don't necessarily reach the threshold for launching an outbreak investigation here in Canada. But knowing that there's an issue going on in the US might prompt us to look more deeply into these matches in Canada, because we know that this could signal a potential outbreak. And especially if it is a produce-related issue, we want to be able to get ahead of it just because it's a very perishable item, right? And from the epidemiologic perspective, we do have recurring strains. So there's some, outbreak investigations that we tend to investigate year after year, the same genetic strain popping up. So we're definitely on high alert when those outbreaks pop up again. We have a good idea of what kind of things we should be looking for, whether it be leafy greens or, something different. And then also with produce-related outbreaks, one of the biggest challenges is getting sufficient detail at the investigation stage. So you can ask someone about, the salads that they ate, 4 or 6 weeks ago, but they may not remember the details of what brand it was. They definitely will not remember what the best before date was on the packaging. So we're trying to get that message out there to investigators at the frontline level when they do their first outbreak follow-up, and usually that's around a week or two after they're, diagnosed with the disease, is to put into practice asking more detailed questions and collecting more detailed information at that initial stage when, it's more likely to be fresh in people's heads. and they're more likely to have packaging available. Getting that information up front can help us, a few weeks down the road when we eventually identify that they're part of a larger outbreak.
Yeah, for sure. That's all really important. Lawrence, I'm not sure if you wanted to add anything to that, or?
Yeah, actually. So in addition to everything Joyce said, another reason why there's delays in outbreak identification is because most people who have a foodborne illness, in order to have a sample taken and tested by the lab, that the sick individual actually has to go to a healthcare professional, and most people don't do that. And so we know that, for example, for every one person that's infected with the Salmonella or E. coli or Listeria, there's others, typically between 20 or 30 people who we don't know about. And so that can also delay the identification of the outbreak, which, as Joyce said, compounds issues because people can't remember what they ate, weeks ago. So I'm leading a large project in collaboration with the Public Health Agency of Canada to actually come up with a new way to identify outbreaks, hopefully faster. And the idea is to actually test wastewater, sewage wastewater, in geographical regions like cities, for the presence of using genomics for the presence of pathogens. And so if we see a spike in the concentration over what is normally there, that could indicate that there are people who are sick and with that pathogen, which could indicate an outbreak. And so we're seeing this actual technique. It's called wastewater-based epidemiology. We're seeing it used right now in the current COVID pandemic, where multiple jurisdictions are doing that, and including myself. And we're giving the data to the public health officials, and they're using it. So, if this was done continuously, then instead of perhaps 3 to 5 or 6 weeks before an outbreak is known, perhaps we could identify an outbreak within a few days of the beginning of the outbreak. One of the limitations of this, though, is that we still don't know who is infected. We would know in a city there are people who are infected, but we don't know who. So to solve that, we're combining this wastewater-based epidemiology with what's called social media syndromic surveillance. And that's where one can look at social media. People may not go to a hospital when they're sick with a foodborne illness, but they like to talk about it on social media. And so in the United States, for the past 7 or 8 years, there are many jurisdictions that have employed social media syndromic surveillance, where they look for symptoms consistent with a foodborne illness in social media to identify outbreaks. And it has been working. So our idea would be to combine that with the wastewater-based epidemiology. And this would potentially allow us to be in contact with those people within the same region where we're seeing the increased wastewater signal, who are also talking about symptoms consistent with the pathogen that we're seeing. And in that way, if we were able to do that within a week, for example, of seeing an increase in the shedding in wastewater, and interview those people, they have a much better chance of remembering, what they ate versus if that interview were to happen 6 weeks later.
Oh, for sure. That's really exciting. I know we've seen a lot of the wastewater testing being done during COVID. So it's quite interesting, and it's exciting to see some use of it in this type of situation as well. So that looks like all the questions we've got for today. So that's great. I'd like to thank both of you for being our panelists and for joining us today, and for all the great information and discussion that we've had. It's been fantastic. So that's all we have. I think, I just wish and hope everyone enjoys the remainder of Fresh Week. And thanks for joining us.
Thank you to our panelists, and thank you to our listeners for joining in. Until next time, fill half your plate with fruits and veggies, continue to seek out new knowledge, and basically never stop growing.